So in this video lecture, we will study about the restriction enzyme. First of all, we will see what are restriction enzymes. So restriction enzymes are basically, these are molecular scissors that cut the double stranded DNA molecules at a specific sites. This is found naturally in the bacteria and various prokaryotes. This is an important tool for manipulating the DNA. Now, what is the basic role of the restriction enzyme in bacteria and prokaryotes? So most bacteria uses restriction enzyme as a defense against bacteriophages. As you know, bacteriophages eats down the bacteria. So this restriction enzymes is used as a defense tool against bacteriophages. So restriction enzymes prevents the replication of the phages by cleaving its DNA at specific sites. The host is protected. The restriction enzyme itself does not cause any harm to the host that is infected by the bacteriophages, that is bacteria, because it is protected by its DNA is protected by the methylase, which adds methyl to the adenine or cytosine bases within the recognition site, thereby modifying the site and protecting the DNA. Now, restriction enzymes are basically molecular scissors. So, what is the history of restriction enzyme? So, first restriction enzyme isolated was in 1970, that is HIN3, and there was subsequent discovery, characterization of various restriction endonucleases. From over 3000 restriction enzymes have been studied in detail, and more than 600 of these are routinely used in lab commercially. So what is the mechanism of action of the restriction enzymes? Restriction endonucleases basically scans the length of the DNA and binds to DNA molecule when it recognizes a specific sequence and makes one cut in each of the sugar phosphate backbone of the double helix by hydrolyzing the phosphodiester bond. Specifically, the bond between 3' O atom and the P atom is broken. Now, direct hydrolysis by nucleophilic attack at the phosphorus atom takes place. So, 3' OH and 5' phosphate is produced, as you can see in the figure. Magnesium is required for the catalytic activity of the enzyme. It holds the water molecule in a position where it can attack the phosphoryl group and also help polarizing the water molecule towards deprotonation. Now, what is palindromic sequence? So, in order to understand the nature and functioning of the restriction enzyme, understanding palindromic sequence is very important. The mirror-like palindrome in which the same forward and backward are on single strand of DNA strands, as in GTT ATG. As you can see, the inverted repeat palindrome is also a sequence that treats the same forward and backward, but the forward and backward sequences are found in complementary DNA strands, like GTA-TAC being complementary to CA-TA-TG. Inverted repeat palindromes are more common and have greater biological importance than mirror-like palindromes. So as you can see, this is GAATTC and CTTAAG. Now, what ends does restriction enzyme fragments restriction enzyme generates? So basically, these the ends of restriction fragments are two types, that is blunt end and sticky end. So blunt ends are some restriction enzymes cut DNA as opposite bases. They leave blunt end DNA fragments. The blunt end fragments can be joined to any other DNA fragment with blunt ends only. Enzyme useful for certain types of DNA cloning experiments are basically create blunt ends. Sticky ends. Most restriction enzyme makes staggered cuts. Staggered cuts produce single-stranded sticky ends. You can see in the figure. So a restriction cleavage site is there, and it produces a sticky end. Now, sticky ends are useful. DNA fragments with complementary sticky ends can be combined to create a new molecules, which allows the creation and manipulation of DNA sequences from different sources. Now, what are isosomers and neosomers? Restriction enzymes that have same recognition sequences as well as same cleavage site, it is called isosomers. These enzymes are mostly used in labs. and restriction enzyme that have same recognition sequence but cleave at different site within that sequence are called neosisomers. As you can see, two examples are there. First one is SmaI, which is which is uh, having the XMA1, which is basically the isosisomer, and another one is SMA1. This is neosisomer. Now, nomenclature of the restriction enzyme. Each enzyme named after the bacterium for which it is isolated using a naming system based on bacteria, genus, species, and strain. For example, EcoRI, where E indicates, abbreviation E indicates Escherichia, description its genus, and co indicates meaning coli, and description is species. R is RY13, that is strain, and I is the first identified order of the identification in the bacterium. Now, types of restriction enzymes. Restriction endonucleases are categorized into three general groups: type 1, type 2, and type 3. So, these types are categorized based on their composition, enzyme cofactor requirement, the nature of their target sequence, position of their DNA cleavage site relative to the target sequence. Now, what are type 1 restriction enzyme? Type 1 restriction enzymes are capable of restriction and modification activities also. The cofactor as adenosine, methanol, adomethionine, ATP, and magnesium are required for their full activity. Contain 2R restriction subunits, 2M methylation subunit, and 1S specificity subunit. This cleaves DNA at random length from recognition sites. Now, type 2 restriction enzymes, these are most common restriction enzymes available in used restriction enzymes. They are composed of only one subunit. The recognition sites are usually undivided and palindromic and 4 to 8 nucleotides in length. They recognize and cleave DNA at same site. They do not use ATP for their activity, but they usually require magnesium as a cofactor. Now, what are type 3 restriction enzyme? These recognizes two separate non-palindromic sequences that are inversely oriented. They cut DNA about 220 to 30 base pairs after the recognition site. This enzyme contains more than one subunit and require adomethionine and ATP cofactor for their roles in DNA methylation and restrictions. Now, what are type 4 restriction enzymes? They cleave only normal and modified DNA, methylated and hydroxymethylated and glucosyl hydroxymethylated bases. Recognition sequence have not been well defined. Cleavage sites takes place basically 30 base pair away from one of the sites. Now, what are the applications of restriction enzyme? They are used in gene cloning, protein expression experiments. Restriction enzymes used in biotechnology to cut DNA into small strand in order to study fragment length, differences among individuals, restriction fragment length polymorphism also to identify particular suspects and criminals. Each of these methods depend on the use of agarose electrophoresis for separation of the DNA fragments. Now, what is RFLP? RFLP is a difference in homologous DNA sequences that can be detected by the presence of the different lengths after digestion of the DNA samples. The method of DNA analysis by RFLP. The method of DNA analysis by RFLP involves the following step. In the first step, fragmentation of a sample of DNA is done by restriction enzyme, which can be recognized and cut whether a specific short sequence occurs in a process known as restriction digestion. The resulting DNA fragments are replaced by the length through a process known as agarose electrophoresis, then transferred to a membrane via the Southern blotting procedure. Hybridization of a membrane to a labeled DNA probe will be done, and it determines the length of the fragments which are complementary to the probe. Then we will observe the fragments of different length, and RFLP occur when the length of a detected fragment varies between individuals. Each fragment length is considered an allele and can be used in genetic analysis. So this is all about restriction enzyme in this video. Hope this video is very helpful to you. If you have any queries, please write in the comment section. I will be very happy to answer your questions. And please like and subscribe the channel. I need your support, and we'll bring more videos like this so that your concept will become more clear, and I will try to be becoming more creative with my videos. Thank you. That's all.